Well, hello, ladies and gents. Happy today, whatever day that may be. Hope you're doing well. So let's just jump right in and talk about membranes. Membranes, so important, they get their own channel, chapter. And maybe someday you could be a television producer and have your own membrane channel. It could compete with the Oprah Winfrey Network. Membranes, what's the big deal? Well, a key feature of membranes is that they form this sort of semi-permeable barrier that allows some materials in and some materials out. A key feature of membranes is that they can create this environment inside of a cell that's fairly regulated and can sort of have its own metabolism, have its own set of chemical reactions that, again, it's not completely isolated from the rest of the environment, but it can concentrate certain enzymes and other molecules and perform certain reactions. So life doesn't happen without membranes. And again, it's semi-permeable, which means some materials can cross while others can't. Now, just sort of a good sort of general rule of thumb of how to conceptualize this, like dissolves like, right? So if it's small, uncharged, or small nonpolar, it can pass through the membrane. Things that are, again, small, nonpolar can pass through. Now, I know they have water listed there, but there are channels in membranes that make water move through rapidly. So water doesn't just fit between the phospholipids easily on its own. But some molecules can pass through much more readily than others. Now, if it's large, charged, and polar, then it has trouble fitting through the membrane, and so it's going to need some help. And we'll talk about that help more later on. All right, so what makes membranes behave the way they do? It's phospholipids. Now, recall we said phospholipids are amphipathic. They have a head that has a phosphate group, and it's got a phosphate or a phosphorus, a bunch of oxygens. So the head is polar, so it will be oriented towards water. And then the tails are hydrocarbon tails, and they're nonpolar. Now, this image on the right side of your screen is pretty good because what it does is it shows how the tails are kinked. And because the tails are kinked, they stay pushed apart from one another. And because the phospholipids are pushed apart, it actually stays fluid. So our membranes are made of phospholipids, but those phospholipids aren't static. The phospholipids are actually moving around one another. So because the phospholipids have those bent tails, again, they stay pushed apart a little bit, and that allows them to sort of dance or twirl around one another within that's bilayer. And that's important because having a fluid membrane allows you to do things like this. Yeah. Oh, I'm sorry, my dog is feeling hostile towards membranes. Hey, come on, guys. That's not cool. But again, by being able to bend membranes, it happens because of the shape of these hydrocarbon tails and phospholipids. The mosaic part of it is due to all the proteins that are embedded in the membranes. And again, we'll discuss those a little bit later. All right. So again, the idea is to imagine those phospholipids sort of, I think of it, it's a little weird, but I think of like waltzing or like, Beauty and the Beast, the big waltz, the big dancing scene. They sort of twirl around one another. And it's only occasionally that they flip bilayers. But for the most part, those phospholipids are moving around one another. So what can happen is small nonpolar molecules can actually squeeze between the phospholipids and get through the membrane. But again, larger molecules are going to have trouble doing that, or stuff that's polar or charged is going to have trouble doing that. Now, embedded in the membrane are proteins, and those proteins sort of float around within the membrane, or some of them are anchored, sort of like a buoy in the water. So it's sort of wild to think about. But our membranes are like the consistency of vegetable oil. If you take olive oil or vegetable oil and rub it between your fingers, that's about the consistency of a cell membrane. And so it's sort of crazy that something that's only 2 molecules thick that are fluid and moving around is what holds us together and allows life to be created. So it's really pretty spectacular. All right. Membranes are subject to the same, forces of fluid dynamics as anything else. So membranes can have their properties affected by temperature. When temperature cools, there's less kinetic energy in the system, right? So the phospholipids tend to pack in more tightly. And when that happens, it can make it more difficult for materials to move through the membrane. So what some species of organisms have actually evolved the capacity to do is to change the production of phospholipids. So, the smoothie R is a membrane factory. So they can actually crank out phospholipids that have more double bonds, and that keeps the phospholipids pushed apart so they can maintain their fluidity at lower temperatures. So it's really spectacular, sort of the capacity to change membrane viscosity just by changing those hydrocarbon tails. All right, now cholesterol is another structural feature in membranes. Remember, cholesterol is 4 fused rings of hydrocarbons. So you see the cholesterol is embedded in the membrane. Now, cholesterol helps at temperature swings or temperature extremes. If you heat up a membrane, the phospholipids are going to be moving around more rapidly, and it destabilizes the membrane. So what I think of cholesterol as being in those circumstances is sort of like an administrator, like a principal or vice principal standing in the middle of a hallway. If they're standing in the middle of a hallway during a passing period, it's going to slow down the movement of people through the hall. Well, if you have these cholesterols, they're like little biological rebar. If you have those embedded in the membrane, under higher temperatures, it's going to slow down the movement of the phospholipids and keep the membrane more stable. Now, on the flip side of that, when temperatures drop, phospholipids can tend to pack in, but what the cholesterol does is it keeps the phospholipids apart so that they don't pack in too tightly at lower temperatures. I think of that as being like a chaperone at a dance, being like reminding people to maybe have a little daylight in between you. Again, cholesterol works at either temperature extreme. It slows down the movement of phospholipids at higher temperature, and it keeps the phospholipids from packing in too tightly at lower temperature. So cholesterol is actually an important structure found within the bilayer. And of course, it's in the nonpolar portion, right? So the hydrocarbon tails are in the middle, and that's where the cholesterol will be found. All right, so there are all sorts of proteins embedded in membranes. So we'll run through that because the proteins are what help determine a lot of the functions of cells and cell membranes, because proteins can do all sorts of different jobs. All right, so proteins can be categorized in different ways. Peripheral proteins are found on the surface of cells, whether it's an interior surface or an exterior surface. And a lot of times those can be used for interactions with other substances or relaying messages inside of cells. Integral proteins actually span the bilayer. So integral proteins are embedded within the phospholipid bilayer. So think about what you already know about a protein like that. If you look at that, these portions of the proteins, these domains that are the helices, what could you guess about their R groups? Exactly. Their R groups are nonpolar or hydrophilic. So nonpolar amino acids will make this integral protein stable in the membrane, embedded in the membrane. But on the ends, here, the loops that are on the exterior or interior of the membrane, you can guess that those R groups are hydrophilic. So again, the proteins are stable because of the R groups in the amino acids that make up the protein. Certain domains or certain regions within the protein will have nonpolar R groups, so it'll be stable in the nonpolar part of the membrane. And then other R groups will be hydrophilic, so they'll be stable on the external part of the membrane, or I should say on the interior and exterior surface of the membrane. Now here's a cool example of sort of the importance of these proteins that can serve as receptors or docking stations on the surface of cells. Here, again, you can have membranes on the surface of the cell. The CD4 receptor is a really important protein for HIV infection. The virus has these spiky molecules on the surface, and what happens is that HIV virus, its membrane has to have its proteins dock on this combination of a CD4 receptor and the CCR5 receptor. So if both those proteins are present, it allows the virus to dock, the membrane to open up, and the virus to dump its contents and infect the cell. Now, what has been found is there are certain pockets of people around the world that are resistant to HIV. They won't be infected with the virus. And so when they've done testing on these individuals who are resistant to HIV, what they found is these people don't produce this CCR5 co-receptor. So if you lack the co-receptor, the virus may be able to dock on the CD4 receptor, but it can't infect the cell. So being able to maybe block the co-receptor, that CCR5 receptor, could help be a way to prevent HIV infection in cells. Okay, so sort of understanding the importance of these different proteins allows you to develop sort of different therapeutic interventions. So here's a nice image from the book looking at some of the different types of things proteins can do. Proteins can work in translocation, that's moving materials in or out of the cell. So you can have channels, or you can have what are called carrier proteins or pumps. So that allows you to move materials, again, in and out of the cell. Sometimes proteins embedded in the cell can act as a way to concentrate enzymes for a series of chemical reactions. This is a common feature in like bacterial cells because they don't have organelles. They have clusters of proteins inside their membrane where you can have sort of chemical reactions occurring. Signal transduction, this is going to be in the next unit. This is a big, or in a couple units, this is a big part of how communication occurs. So proteins can actually cause shape changes that cause communication within cells. Cell-to-cell recognition, this is a pretty cool one. On the surface of your cells, you have little sugars or carb chains. So you see the little green sugar on this glycoprotein, gly, and protein. It's a sugar attached to a protein embedded in the cell membrane. What's cool is every one of your cells has a glycoprotein that's unique to you. It's like a little molecular ID badge that's on the surface of every one of your cells. So cells in your immune system can look at these glycoproteins and know, oh, that's one of my person's cells. So if a cell shows up that has different proteins or glycoproteins on its surface, then your body can recognize it as not belonging to you and try to get rid of it. This intercellular joining, again, it allows sort of stability in tissues. And then attachment to the exoskeleton. So again, what can happen is you can have proteins and sugars sort of enmeshed around the surface of the cell, and that holds cells in place or stabilizes membranes. All right, so role of membrane carbs in cell-to-cell recognition. So there are molecules in the surface of cells that, again, allow for communication between cells. So a good example is blood types. This is one they like to talk about on the AP exam frequently. So if you look at the surface of a cell, there could, again, be these carb chains on the surface. And what happens with our blood cells is they have certain combinations of molecules on their surface. So for instance, if you have type A blood, that means you have the A marker on the surface of your blood. So if we look here, A blood means you have the A marker, which looks like it's blue. B blood means you have the B marker, which in this case looks green. And then if you have AB blood, that means you have both protein or both markers. You have the A marker and the B marker. And then having type O blood, type O means you don't have any of those markers. So when they talk about blood types A and B being codominant, that means both proteins are being expressed. So the A marker and the B marker are being equally expressed. And then O is recessive because in this case, recessive just means they don't make anything. So type O blood means it doesn't have any of those markers. Type B blood has the B marker. Type A blood has the A marker. And AB blood has both markers. Now, sometimes people ask about like being A positive, B negative, that kind of stuff. The positive and negative is for another type of protein. It's called the rhesus factor or RH factor. So that's something that was found in rhesus monkeys. So that's how I got that designation. But if you're RH positive, that means that protein is present. And if you're RH negative, that means that protein isn't present. So like, for instance, if you have type O negative blood, if you're O negative, you don't make any of those proteins. So there's nothing on that surface of the cell. If you're O positive, you don't have any A or B markers, but you have that RH factor. Now, if you're B negative, you have the B marker, but you don't have the RH factor. If you're B positive, you have the B marker and the RH factor. And then if we look over here, if you're AB positive, you have the A marker, the B marker, and the RH factor. So you get a lot of information that you can glean just from the designation of the naming of the cell type. All right, let's see. So again, this is just sort of going what can pass through the membrane and what can't. So remember, you have that non-polar middle portion of the membrane. So that's the gatekeeper. If something is large, charged, or polar, it's not going to pass through the non-polar part of the membrane. The stuff that can pass is stuff that's small and non-polar. Okay. Let's see. So I just saw something there. Sugars, because they're polar, with all that oxygen, like glucose here, it has trouble passing directly through the membrane. So you're going to need a protein to help that. All right. Looking at one particular type of protein, there's what's called a protein channel. So a protein channel is like a little tube or a straw embedded in the membrane. So what can happen is you can have certain chemical properties of the interior of that channel that allow certain substances to pass. So certain channels only allow certain materials. You could have a chloride ion channel, you could have a potassium ion channel, and they only allow certain substances to pass through them. Aquaporin is one such channel. Aquaporins allow only the passage of water. So again, you can imagine there are going to be hydrophilic portions on the interior of that protein that allow the water to pass through. So again, aquaporins, water won't easily pass between phospholipids, but cells will have aquaporins in their membranes to facilitate the rapid movement of water. Here's another important example of a channel protein. It's the CFTR protein. It's like cystic fibrosis transmembrane conductance receptor protein. So there's this protein that's in the membrane of cells, and normally it allows chloride ions to flow out. Now, if a person inherits a certain version of the gene for making that protein, you can change the amino acids. And if you change the amino acids, it'll fold up differently. So there you can see a functioning CFTR protein channel, and here's a non-functioning protein channel. You change the shape, right? When you change the shape, it can block chloride ions from leaving the cell. Now, if the chloride ions don't leave the cell, think about what that's going to do to the solute concentration in the cell. Solute concentration will go up. And if solute concentration goes up, water potential goes down. And water flows from high to low potential, right? So if you have a non-functioning CFTR protein, the water potential will drop inside the cell. Water will flow into cells. And then what you have in like the lining of the respiratory tract and digestive tract is mucus that is normally really watery and can allow materials to pass through fairly easily. The mucus becomes really thick and sticky. Now, in your respiratory passageways, if you have that thick, sticky mucus, when you breathe in, that allows pathogens to dock and not be swept out by the cilia lining the respiratory tract. So that can cause lung infections and damage to the lungs. And in the digestive tract, that thick, sticky mucus can block glands in the pancreas from releasing digestive enzymes. So you can have issues with digesting food adequately as well. So cystic fibrosis has really profound impact on a person's health because you basically block the flow of ions, and that changes water potential. So, the stuff we're learning has real life application. It has real life impact on a lot of people. All right, carrier proteins. Carrier proteins are different from channel proteins because a channel protein is like an open tube, like a straw. Carrier proteins are a little different. They'll actually change your shape. So let's watch what's going on in the clip here. Too big, not going to work. If you look, it doesn't quite fit there. Doesn't fit there. Oop, pass through the membrane must be small, nonpolar. That, nope. Okay, look what happens here. Whoop. The carrier protein changed its shape, right? So that's the key feature of a carrier protein. A carrier protein will change its shape, and when it changes its shape, that translocates or moves the solute across the membrane, okay? So again, the key feature of carrier proteins is that they'll actually change their shape to move the material through the cell. All right, have a quick clip here looking at some of these membrane proteins.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it.
Now, I like the fact that this is looking at the fluidity of the membrane. The one downside with this animation, and something I've not been able to find in any animation, is it doesn't show how those phospholipids are actually waltzing around one another, twirling around one another. Okay, so let's listen to this. I think the music is quite inspiring. It sounds like you're swimming underwater in the ocean.
Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals.
So in the middle there, that looks like a channel. So you can see how the channel is open and allows substances to pass through. Over here, I think this is a carrier protein. It will actually change its shape.
From the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
All right, good. Let's please cancel that. Oh boy. All right, internet.
All cells have a...
Okay. So a nice view of some of the work that proteins can do. All right, so now let's actually pause there. Done with the stuff on membranes. We'll look at transport here in another video clip. Thanks a lot.